Welcome to Biochemistry DSP channel. In today's topic, I'll be discussing about the metabolism of ketone bodies. Ketone bodies. Acetone, acetoacetic acid, and beta hydroxybutyric acid. These are collectively called as ketone bodies or acetone bodies. These ketone bodies are synthesized in the liver and they are water soluble, and these ketone bodies are also used as an alternative fuel in the tissues, extrahepatic tissues such as skeletal muscles, cardiac muscles, renal cortex, etc. They are used as an alternative fuel. Uh, During certain metabolic, physiological, and pathological conditions like diabetes mellitus, renal glycosuria, and during starvation condition, and during liver damage, when there is low dietary carbohydrates, so during that insufficient amount of dietary carbohydrates, beta oxidation of fatty acids will be accelerated in the liver. Due to beta oxidation of fatty acid, more production of acetyl CoA uh, will be available, and this acetyl CoA will not be fully utilized by the liver through TCA cycle. During that condition, due to the inability of the liver to uh, oxidize the excessive ketone acetyl CoA, it will divert into a pathway called ketone bodies formation or ketogenesis. So ketogenesis is nothing but synthesis of ketone bodies. Okay, synthesis of ketone bodies like acetone, acetoacetic acid, and beta hydroxybutyrate is called as ketogenesis. The major site is liver is the only organ which can synthesize ketone bodies, and this synthesis of ketone bodies takes place in the mitochondrial matrix of the hepatic cells. The condition which favor ketogenesis includes two conditions. Okay, the during uh, actually in the normal healthy person, liver will constantly produce ketone bodies in small quantity. So the minimum concentration of ketone bodies production will be less than 3 milligram per 100 ml in the blood. Okay, so when this ketogenesis will increase means under two main conditions: when body fails to utilize the available carbohydrate, so like blood glucose or hepatic glycogen, or okay, when the body fails to utilize the available carbohydrate like blood glucose or hepatic glycogen, that is one condition. The other condition is where dietary carbohydrates are not available as a source of metabolic energy and hepatic glycogen is completely exhausted. So during these two conditions only, the synthesis of ketogenesis will be seen more. Okay, the conditions includes the metabolic, physiological, or pathological conditions like diabetes, mainly type 1 diabetes mellitus, renal glycosuria, prolonged starvation, and glycogen storage disorder. So during these conditions, when there is low amount of carbohydrate diet, fat will be acting as the source of fuel. So beta oxidation of fatty acid will be accelerated in the liver, which will leads to production of more amount of acetyl CoA. The level will exceed the capacity of the liver to convert this to oxidize this acetyl CoA via TCA cycle. So liver will convert the excess of acetyl CoA into ketone bodies through ketogenesis. So the reactions which occurs in ketogenesis involves three steps. First step is the formation of acetyl CoA. Okay, two molecules of acetyl CoA condense. Two molecules of acetyl CoA will condense to form the first product, acetoacetyl CoA, in the presence of the enzyme thiolase. Then acetoacetyl CoA will condense with one more acetyl CoA in the presence of the enzyme HMG CoA synthase to form the other product HMG CoA. HMG CoA is nothing but beta hydroxy beta methyl glutaryl CoA. Then this beta HMG CoA 3-hydroxy 3-methyl glutaryl CoA, that is the HMG CoA, will undergo cleavage to yield two products, acetyl CoA and acetoacetate. Okay, so from the cleavage of this HMG CoA, we get acetoacetate as well as acetyl CoA through the help of the enzyme HMG-CoA lyase. So HMG-CoA lyase is the enzyme which will convert HMG-CoA into acetoacetate and acetyl-CoA. So the first ketone body acetoacetate is produced. Okay, so in the ketogenesis process, the first ketone body produced is acetoacetate. It is produced by cleavage of HMG-CoA with the help of the enzyme called HMG-CoA lyase. Then from acetoacetate, this acetoacetate is reversibly reduced. Okay, it will be reversibly reduced into beta hydroxybutyrate with the help of the enzyme called beta hydroxybutyrate dehydrogenase. It's also called as 3-hydroxybutyrate dehydrogenase, or it can be spontaneously decarboxylated into acetone. So from this acetoacetate, we get the secondary ketone bodies acetone and 3-beta hydroxy butyrate. So other ketone bodies are synthesized from acetoacetate. So one way of production is by spontaneous decarboxylation. Okay, acetoacetate will undergo spontaneous decarboxylation to yield the compound acetone, or it can be reduced into beta hydroxybutyrate with the help of the enzyme beta hydroxybutyrate dehydrogenase. So this is the pathway which occurs for the production of the three ketone bodies acetoacetate, acetone and beta hydroxybutyrate. So these are the three steps which are involved to form acetone, acetoacetate and beta hydroxybutyrate. Regulation of this ketogenesis. So regulation involves excess, that is in the adipose tissue, due to excess production lipolysis will occur in the liver, so that excess of triacyl glycerol will be converted into free fatty acid. These free fatty acids will uh, enter through blood into liver, where it will be undergoing beta oxidation to be converted into acyl CoA. Then from this acyl CoA, either it can be this fatty acid, fatty acyl CoA can be esterified to form acyl glycerol, or it can undergo beta oxidation to form acetyl CoA. This acetyl CoA will enter into ketogenesis pathway to produce ketone bodies and CO2. So acetyl CoA, okay. So formation of this acetyl CoA will be a key regulatory mode to control this ketogenesis production. So, okay, the significance of this ketogenesis pathway. So, ketone bodies serve as an alternative fuel for the heart, renal cortex, and skeletal muscles. When the body fails to utilize carbohydrate as the available source of energy, that occurs during diabetes mellitus, renal glycosuria, and during starvation, when the glycogen storage in the body is completely depleted. In tissues which lacks mitochondrial cells such as sorbecies, renal medulla, cornea, and islands cannot utilize these ketone bodies as an alternative fuel, because oxidation of ketone bodies takes place in the mitochondria. So the oxidation of that is utilization. Okay, the utilization of ketone bodies will occur only in the mitochondria. So these cells which lack mitochondria will not able to utilize this ketone bodies as a secondary source. So in the well-fed state, the concentration of ketone bodies in the blood will does not exceed beyond 3 milligram per dl. So due to overproduction of ketone bodies in the uncontrolled diabetes or during prolonged starvation, it will exceed the normal rate 3 milligram per dl. So when the rate of production of ketone bodies exceeds the rate uh, of their use by extrahepatic tissues, their concentration also increase in the blood. So increase in concentration of this ketone body in the blood is called as ketonemia. So due to ketonemia, excess of this ketone bodies will be excreted in urine. That condition is called as ketonuria. So from ketonuria, you get ketonuria. During that ketonuria condition, the urinary excretion will increase as high as 5000 milligram per day. Normally, ketone bodies will be excreted in the amount of 90 milligram per dl, whereas during abnormal condition, it may go as high as 5000 milligram per day. So this presence of these ketone bodies in urine can be detected using Rothra's test. Since acetone is a volatile compound, it can be easily exhaled through lungs, imparting a fruity smell to the patient's breath. So this will be a helps to diagnose the condition of ketonemia in diabetic patients. Then the condition that may lead to ketosis or prolonged starvation when the glycogen storage in the body is totally exhausted during type 1 diabetes, renal glycosuria, and diet low in carbohydrate but rich in fat, and severe exercise. Okay, so severe exercise, renal glycosuria, low carbohydrate diet but rich fat diet, glycogen storage disorders, type 1 diabetes, all this, and prolonged starvation. So all this will lead to a condition called ketosis. And ketogenic substances means it includes free fatty acids, ketogenic amino acids, and increased glucagon hormone and stress hormone like epinephrine, norepinephrine. So all these are called as ketogenic substance which promote ketogenesis. So the substance which can promote ketogenesis are free fatty acids, ketogenic amino acids, and hormones like glucagon, epinephrine, and norepinephrine. Then anti-ketogenic substances. So substances like carbohydrate, that is glucose, then glucogenic amino acids, glycerol, and insulin. Okay, mainly glucose, glucogenic amino acid, glycerol, and insulin act as anti-ketogenic substances that will inhibit ketogenesis. Next is the utilization of ketone bodies in the extrahepatic tissues. So, in the extrahepatic tissues, almost all the extrahepatic tissues, uh, excluding which lack mitochondria, will undergo the oxidation of ketone bodies. So, oxidation of ketone bodies is nothing but utilization of ketone bodies. It's called as ketolysis. So, lysis means breakdown of ketone bodies. So, this breakdown of ketone bodies occurs in the mitochondrial matrix. So the reactions which takes place in the mitochondrial matrix in ketolysis includes oxidation of this beta hydroxybutyrate. So the first step is oxidation of beta hydroxybutyrate into acetoacetate. The beta hydroxybutyrate is oxidized to acetoacetate in the presence of the enzyme called beta hydroxybutyrate dehydrogenase. Second step is activation of acetoacetate into acetoacetyl CoA. Okay, so this is a main key step. So in this conversion of acetoacetate into acetoacetyl CoA, the donor of this coenzyme A is succinyl CoA. So succinyl CoA will be converted into succinase, and acetoacetate will acquire that coenzyme A to be converted into acetoacetyl CoA in the presence of the enzyme called thiophorase. It's also called as succinyl CoA acetoacetyl CoA transferase. Okay, so the enzyme thiophorase uh, is present only in the extrahepatic tissues, but it is absent in the liver. So hence liver cannot undergo ketolysis reaction. So you have to understand why synthesis, okay, synthesis of ketone bodies occurs in the liver. Why not utilization also can take place in the liver means this is the reason, because the activation step of acetoacetate into acetoacetyl CoA occurs only in the presence of the enzyme called thiophorase, which is present only in the which is not present, okay, which is totally absent in the liver. So liver lacks this enzyme, this mitochondrial enzyme thiophorase succinyl CoA acetoacetate CoA transferase. So it is absent in the liver. Therefore, liver cannot utilize this ketone bodies. Liver cannot undergo ketolysis reaction. Hence, it occurs in the extrahepatic. That's the reason why it occurs. Ketolysis occurs in the extrahepatic tissues because of the lack of this enzyme thiophorase. So now acetoacetate is converted into acetoacetyl CoA in the presence of the enzyme thiophorase. Then cleavage of this acetoacetyl CoA into acetyl CoA. So in the presence of coenzyme A, the acetoacetyl CoA will be cleaved into two products. One is it will be cleaved into two molecules of acetyl CoA. Okay, so acetoacetyl CoA will be cleaved with the help of the enzyme called thiolase, and it will be converted into two molecules of acetyl CoA, which will enter into TCA cycle to provide energy. The acetoacetate, okay, thus the three ketone bodies beta hydroxybutyrate, acetoacetate are reconverted into acetyl CoA, and they enter into TCA cycle, which can be oxidized to CO2 and water to provide energy. Since acetone cannot be easily oxidized, okay, acetone is easily exhaled through lungs because of the volatile nature, and it gives a characteristic fruity smell to the patient's breath. Okay, so even though liver produces keto bodies, that is ketogenesis occurs in the liver, but liver cells cannot utilize ketone bodies as a source of fuel due to the lack of the enzyme called thiophorase. This is the reason, okay, why liver cannot utilize ketone bodies as a source of fuel means it lacks the enzyme thiophorase. So in the extrahepatic tissues, okay, uh, whereas okay, liver lacks the enzyme thiophorase. The same way, the extrahepatic tissues also cannot synthesize ketone bodies because of the lack of the enzyme HMG CoA synthase. So for synthesis, we need HMG CoA synthase. Okay, for synthesis of ketone bodies, the HMG CoA synthase enzyme is required, which is absent in the extrahepatic tissues. Whereas for ketone bodies utilization, we need the enzyme thiophorase, which is absent in the liver. So, ketogenesis will occur only in the liver, and ketolysis will take place only in the extrahepatic tissues like skeletal muscles, brain, and other tissues. Okay, so and other tissues or cells which lack this mitochondria cannot utilize this ketone bodies as their fuel because uh, this ketolysis occurs in the mitochondrial cells. Since they lack mitochondria, they cannot utilize ketone bodies, and they will not use the energy provided by ketone bodies. Acetone, since it's a volatile compound, it can be easily exhaled through lungs. So this is how the ketone bodies are utilized. Okay. Uh, thank you for watching this video. Subscribe and support the channel Biochemistry DSP.